Hi again, everybody. Today we are going to be discussing mitosis and meiosis once again, but today we will be primarily focusing on meiosis. So let's get into it. Now, meiosis is actually quite different from mitosis. Meiosis is solely for sexual reproduction, and meiosis has 4 divisions, whereas mitosis only has 2. It is also important to note that meiosis is about reproducing an organism, not creating more working cells. Meiosis's process occurs in 2 sets of PMATs, prophase, metaphase, anaphase, and telophase, for those of you who don't know, in a row with interphase at the start of both of them. Moving on to meiosis 1, which is the first half of meiosis. We start off with interphase, and during interphase there is cell growth, duplication of chromosomes, DNA replication, and the duplication of organelles. After that, we move on to prophase 1, which is the first stage of meiosis, and this is where crossing over occurs. I'm now going to take some time to explain crossing over, just for those of you who don't know. In a tetrad, there are 2 sets of a like chromatid. Crossing over is when they swap genetics to become 4 unique chromatid. It's really not that complicated. After that, we have metaphase 1, where the tetrads line up at the center of the cell. Then we have anaphase 1, where the tetrads separate and the chromatids move along the spindles to their centromeres. And after that, we have telophase 1, where the nuclear membrane reappears, but now there are 2 separate chromatids. Now onto cytokinesis and interkinesis. Cytokinesis is when the separation of the cytoplasm creates 2 cells, and interkinesis is the period of rest before meiosis 2. Now onto meiosis 2, which is the second half of meiosis. We start off with prophase 2, where the chromosomes condense, the nuclear envelopes disappear, and the spindle fibers form. After that, we move on to metaphase 2, where the spindle fibers attach to chromosomes and the chromosomes line up in the center of the cell. After that one, we have anaphase 2, where the centromeres divide and the sister chromatids move to opposite ends of the cell as spindle fibers pull them away. After anaphase 2, we have telophase 2, where the chromosomes reach opposite poles and the nuclear envelope reappears. Now there is 2 in each cell. And finally, after that, we have cytokinesis, which is the division of 4 haploid cells. There are now 4 gametes. And that marks the end of meiosis. Hi there, everybody. Today we will be discussing mitosis and meiosis. Now, although the names sound similar, they are actually 2 different forms of replication. Now let's get on to mitosis. Mitosis is quite interesting, as it is the process of cell division which will result in 2 daughter cells that have the same kind and number of chromosomes as its parent cell. In this stage, there are a series of basic steps: prophase, metaphase, anaphase, and telophase. The acronym for these is PMAT, P-M-A-T. There are also 2 other steps: interphase, which comes before PMAT, and then cytokinesis, which comes afterward. Let's move on to these phases. Stages of mitosis are very interesting. The first stage is interphase. It occurs before mitosis begins. The cell is preparing to replicate, and the cell spends most of its life here. The next phase is prophase. Chromosomes condense, the nuclear envelope disappears, the centrioles move to opposite sides of the cell, and the spindle forms and attaches to the centromeres on the chromosomes. Now it's metaphase. In metaphase, the chromosomes line up on the equator of each spindle, and the centromeres move to opposite ends of the cell. Now we have anaphase. In anaphase, the centromeres divide. Each 2 chromatid become 2 one chromatid, and the spindles pull chromosomes to opposite poles. Now we have telophase. In telophase, the chromosomes uncoil, the spindles disappear, and the nuclear envelope reappears, but now there are 2, 1 for each new cell. Now, finally, we have cytokinesis, the final stage. In this stage, the cytoplasm divides into 2 daughter cells, and that marks the end of the process of mitosis.